This is IB Biology, A3.1 Diversity of Organisms. Our two essential questions are, what is a species, and what patterns are seen in the diversity of genomes within and between species? As an introduction, let's take a look at these four species. Here you can see that we have both a common name and a scientific name or a binomial name. These are Latin-based, and they're made up of two words, a genus and a species, and sometimes you'll also see a subspecies. We're going to look now at an example of spinach. Spinach, note, has common names that are different in every language, but the scientific name, this binomial name with two words, genus and species, are going to be the same in every language. And so the benefit of the scientific name is that scientists around the world can use that name and know that they are speaking about the same species without having a language barrier. Note, when you are referring to an organism in an examination, you can use either the common name or the scientific name. IB says either one is acceptable. All right, getting started. Variation between organisms as a defining feature of life. Now, no two individuals are identical in all their traits. The least variation between two organisms is when you have genetically identical twins, known as monozygotic twins, because they started from one zygote, one fertilized egg, that then split and became two individuals. Now, they started with the exact same genetic information, but those two twins have different environmental factors throughout their lives. They're eating different foods, they're getting different amounts of exercise, they're exposed to different toxins, different amounts of sunlight. And so over the course of their life, they become more and more different through environmental factors. Now, patterns of variation are complex, and we're going to see that those form the basis for naming and classifying organisms. Throughout human history, humans have wanted to make sense of our world. We want to know what plants are safe to eat versus not safe to eat, right? Which animals are good to hunt. We want things to be grouped and organized. And the father of this organization was Carl Linnaeus. From the 17th century onward, biologists have used this word species for a group of organisms with shared traits. Like here's a whole bunch of frogs that are green with red eyes. They live in this kind of same environment. Let's call that a species. They're all similar. And so Linnaeus spent his life's work classifying organisms, and he did actually an amazing job. We found, of course, some mistakes now that we can access DNA, but Linnaeus didn't have access to DNA. He didn't even know what it was. And so instead, he was using the morphological species concept. Morphology refers to the physical structure, and so he was just classifying organisms based on how they looked and trying to group them together by organisms that looked similar, that had a particular outer form and inner structure. We use this binomial system I introduced at the beginning of this video. There are two words. There's a couple of rules that you need to be familiar with. So first up, the first word in this binomial name is the genus. A genus is a level of classification above the second word, which is the species. This is an umbrella, and you can have many species within this genus. So it's just one higher level of organization that can contain multiple species. Now, when we write out this binomial name, we always capitalize the genus. We always lowercase the species. When it's typed or printed, we always italicize both words. And lastly, once both words are used one time in a document, written out completely, we can then use a shortened form where we just take the genus name and use that first letter as an abbreviation. So we can just write C. letrons when we're talking about coyotes after both this canis and letrons were used one time. Now, the morphological species concept we talked about earlier has a lot of limitations. Now we are going to focus on the biological species concept. In biology, we define a species, and this is important, little star next to it. A species is a group of organisms that can breed and produce fertile offspring, meaning the offspring are able to make more offspring. So here, for example, we have a horse. If it is bred to another horse, we're going to get a horse. This horse is fertile, and so all of these horses belong to the same species. If a horse, however, breeds with the donkey, they can actually have offspring, which we would call a mule. But mules are infertile. Mules can't make more mules. The only way to get another mule is to breed another horse and a donkey. And so, because even though they can make offspring, they're infertile, horses and donkeys are not considered the same species by the biological species concept. Okay, now there are some challenges to this species concept. For example, captive lions and tigers in captivity have sometimes hybridized, producing ligers or tigons. And in genera, which this word is just plural for genus, in which speciation is occurring rapidly, like certain conifers, there is going to be some interspecific hybridization, and it's less easy to identify species. It can be hard to know, are they able to interbreed and produce fertile offspring? There's always going to be some gray area, especially if speciation is actively occurring. Now, a population is a group of organisms of the same species living in the same area at the same time. So if we have a forest and there's a population of frogs that are living there that are all those green frogs with red eyes, right? That might be a population of a specific species of frogs that are living in that forest at the same time. Two populations that live in different areas can still be the same species, right? There can be another forest a ways away with green frogs with red eyes of the same species, and if they were to meet, they could potentially interbreed and produce fertile offspring. But what we see is that when there's some kind of geographic isolation, we see species starting to diverge. Recognizable differences may develop as the species become genetically more different, and if those differences continue to accumulate, the two populations may eventually become different species as they each adapt to those slightly different environments. And so we call that speciation, the splitting of one species into two or more. It usually happens gradually rather than by a single act, and populations just become more and more different over time. It can therefore be an arbitrary decision whether two populations are regarded as the same or different species. Again, there's going to be some gray area. Are we going to say they're separate species or not? Diversity in chromosome numbers of plant and animal species. Different species have different numbers of chromosomes. You can see here, humans have 46 chromosomes. A good way to remember that, the company that you used to be able to use to do some ancestry testing, where you would take a cheek swab, send in your DNA sample, and they would tell you about where your ancestry is and close relatives. That company was called 23andMe. The reason it was called that is because humans have 23 pairs of chromosomes. You have 23 from mom and 23 from dad. So use that to help you remember that if you add 23 and 23, you're going to get the total number of chromosomes in humans, which is 46. In contrast, we're going to see that chimpanzees have 48 chromosomes. These are really the only two numbers that you should memorize. All of the other ones we're going to look at, you don't need to memorize. All right, so note that while all members of the species have the same number of chromosomes, during evolution, chromosome number can decrease if chromosomes fuse or increase if they split. There are mechanisms that can cause the chromosome number to double. We see that often in plants. However, these changes are rare. Usually there is no change in chromosome number for millions of years. So let's take a look. Here are a whole bunch of different organisms, their number of chromosomes. Now, we use this word diploid and haploid often. Diploid refers to body cells. These are going to be those cells that have all of your DNA, two sets of chromosomes, one from each parent. And so we say 2N because you have two sets of your 23 chromosomes, so a total of 46. Every cell in your body has that except for our sex cells. They are haploid. They only have half the genetic information. Through the process of meiosis, the chromosomes will split in half so that an egg or a sperm cell only contains half of an individual's genetic information. That then allows for two different sex cells to combine and form a whole, a diploid cell. So haploid plus haploid would make a diploid cell. 23 chromosomes in a sperm, 23 chromosomes in an egg. So when they fuse through fertilization, we end up with a diploid cell, which then immediately begins replicating and eventually becomes a multicellular organism. Diploid cells are always going to have an even number of chromosomes due to sexual reproduction. A male gamete and a female gamete fuse to make a new organism. And since both the male and female gamete had the same number of chromosomes, since they're the same species, the resulting doubling will always be an even number. Any number you double, it will always then be an even number. Okay, so you can see here humans with our 46 chromosomes, and it's interesting to compare the number of chromosomes in some other organisms. This here is a karyogram. This is used to study the chromosomes of an organism. Scientists look at cells that are currently dividing, they stain them, and then they photograph them in this image called a karyogram. Now, this is a human karyogram. Remember, we have 23 pairs of chromosomes. Now, our chromosomes are numbered in decreasing size order. So chromosome 1 is going to be the longest. And this is chromosome 1 you got from mom and chromosome 1 you got from dad. Here's chromosomes 2, 3, 4, etc. All the way down to chromosomes 22, which are the shortest. And then that 23rd pair of chromosomes are the sex chromosomes. And we're going to learn about how they're either X and Y or X and X, depending if the individual is male or female. Now, these chromosomes are classified based on 3 types of difference. They're banding patterns. So note that in certain places, the DNA is going to be more densely packed together, and we're going to see banding patterns due to this. There are also different sizes, like we just mentioned, and they also have what's called a centromere. The centromere is the point at which they connect to each other during replication. And so those chromosomes are going to pair up to make sure that the resulting sex cells in meiosis each end up with one of these matching chromosomes. The characteristic types of chromosome in a species are called its karyotype, and the image is then a karyogram. Now, this is a specific example that I'd like you to be familiar with. Here we see human chromosome 2. You can see the banding pattern, and this spot right here indicates the centromere. So this is the point that it would join with another chromosome 2 and pair up. Here we have chimpanzees, chromosomes 12 and 13. Now there's a hypothesis that human chromosome 2 was formed from the fusion of 2 chromosomes in a primate ancestor. So if we compare this chromosome 2 with these 2 from the chimpanzee, we're going to notice that there are some pretty strong pieces of evidence that support this theory. So if you're asked to compare human chromosome 2 with the 2 chimpanzee chromosomes, you might notice the banding pattern of the long arm of chimp chromosome 13 right here is very similar to the long arm of human chromosome 2 right here. Look at that. The long arm of chimp chromosome 12 and the short arm of human 2 have the same banding pattern. Notice. Some bands on the short arm of chimp 13 are missing from human 2. So we don't see these bands here on human 2. And the centromere from chimp 13 is missing from human 2. But otherwise, we see a lot of overlap. So we just here stated some comparisons between those 2 sets of chromosomes. We are then told that the ends of chromosomes called telomeres have many repeats of the same short DNA sequence. If the fusion hypothesis were true, predict what would be found in the region of the chromosome where the fusion is hypothesized to have occurred. And so, if we had fusion right here, and these were highly repetitive sequences called telomeres, what would have happened to those sequences? Well, either telomere could have been deleted, there could still be non-functional telomeres present, or the number of those repeats could have been reduced in order to go from these 2 to this 1. Normally, a chromosome has just 1 centromere, but in chromosome 2, there are remains of a second centromere. Explain this observation. And so here we can see a clear centromere, right, which could have been passed down from chromosome 12, but there could be remains right here from what was originally chromosome 13. Now, of course, these are chimpanzees, but we're saying that some common ancestor of both humans and chimpanzees may have had these 2 separate chromosomes that then fused when humans evolved. And so maybe this centromere was left over and there are just some remnants that we could find as additional evidence. So, discuss the strength of the evidence that there was a fusion of those chimp chromosomes and the evolution of chromosome 2 in humans. The evidence is strong. There's a considerable similarity in banding or sequences, which is very unlikely to be due to chance. Banding patterns from chimp chromosome 12 and 13 are both found in human chromosome 2, and that remnant of a second centromere is even further evidence. Next up, the genome is all the genetic information of an organism, in other words, all of an organism's DNA. Now remember, you have your entire genome in every single cell in your body, with a couple of exceptions, including the sex cells and some exceptions we've already discussed, like red blood cells. But every other cell, a muscle cell, a fat cell, a neuron, they have your entire genetic information. So your 23 pairs of chromosomes are made up of lengths of DNA that are tightly packed, and a length of DNA that codes for a particular protein is going to be called a gene. Now, typically members of the same species have the same genes in the same sequence along each of their chromosomes. So on everybody's chromosome 7, we're going to see that there are genes along this chromosome that are going to control the same factor. Now, there might be some variations in the forms of those genes, but they're going to be at the same location. Diversity within a species is caused by alternative forms of a gene called alleles. Alleles usually only differ from each other by one or a very small number of bases. Sometimes larger sections of a gene become altered, but this usually results in a loss of gene function, which can then lead to inviability. The positions in a gene where more than one base may be present are called single nucleotide polymorphisms. So note, in individual 1 and 2, this DNA sequence is identical other than this one location, where there's a G in one individual and there's an A in another, and so the base pair then, A of course goes with T and G goes with C. This particular location is a location that could form a different form of the protein that would be coded for by that gene, and we call it a SNP, SNP, single nucleotide polymorphism. It's just a location on a gene where we see variation within a species at that particular location. Now, within one individual human, there are typically about 4,000 to 5,000 SNPs out of over 3 billion bases, which is mind-boggling because only about one base in 650,000 is different from that commonly occurring in humans. This just blows my mind about how similar we are. We are almost identical individuals. Only one base in 650,000 is even potentially different from anybody else on this planet. Diversity of eukaryote genomes. Genomes vary in overall size. This is determined by the total amount of DNA, and it can be measured in base pairs. Large genomes can contain a large amount of non-functional DNA, so they don't necessarily contain more functioning genes than smaller genomes. For example, almost half the human genome consists of transposons, most of which have no known function. So here we see genome size in million base pairs. So remember, I just mentioned humans have about 3 billion base pairs, so that is why we see 3,000 here. 3,000 million is the same as 3 billion. Now look at this though. This plant over here, okay, a woodland plant has 150,000 million base pairs. Does that mean that it's way more complicated and complex than humans? It can, play chess and make sentient decisions? No. Again, we're going to see that not all of the genome is actually going to be used. Much of it will be non-functional. And this is an example that I mentioned real briefly earlier, where sometimes commonly in plant species, we'll just see a duplication of the entire genome without then separating cells in two. And so the genome is just effectively doubled, and we end up with, over multiple doublings, very, very large genome sizes. Now, genomes can vary in base sequence. More differences will accumulate over time due to mutation. Variation between species is much larger than variation within a species. We saw that within a species, humans specifically, we have very low levels of variation, but then we have, of course, more variation between species. Now, this is cytochrome C, which is a protein used in cell respiration, which all organisms do to make ATP energy. And so since all organisms make this protein, it's a great comparison protein, and we can look at the amino acid sequences, which are coded for by DNA, and look at the number of differences from humans to be able to build an evolutionary tree and say, how far back did our speciation occur? And you can see here that the chimp has no differences. The cytochrome C in a chimpanzee is identical in amino acid sequence to a human. Now, a rhesus monkey has one difference, but back to a whale now, 10 differences. A turtle, 15. A bullfrog, 18. A tuna fish, 21. A lamprey, 20. And so you can see here that over evolutionary time, the further back we deviated, the more differences there are going to be in our amino acid sequences, which are, of course, coded for by our DNA sequences. We can use genome-sized databases to compare genome sizes between species. Genome size is typically given as nuclear DNA contents of a haploid cell, such as a gamete, and we call that the C value. So that's all of the DNA in the nucleus of an egg or a sperm. Genome size may be given in units of mass, usually in picograms, which is what we're going to look at, or in the number of base pairs or megabase pairs. So your job is to use the genome size database to take a look at the genome size of the following organisms. So we have 4 plants. You'll use this first database to take a look for their C values, and then 4 different animals. I recommend doing a search by the scientific or binomial name. Current and potential future uses of whole genome sequencing. So whole genome sequencing is determining the entire base sequence of an organism's DNA. This was first done in the 1990s, but it was very, very slow and very, very expensive. Today, the increasing speed and decreasing costs of whole genome sequencing has allowed for the genome of thousands of species of organisms to be sequenced, like those databases that you just looked at. For example, it cost $100 million to sequence one human genome in 2001, but the cost today is less than $1,000. The current uses of whole genome sequencing. Why do we want to do this? Number one, we can do research into evolutionary relationships. We can compare genomes to allow researchers to identify relationships between species and trace the diverging pathways from common ancestors, which can make it easier to conserve and protect biodiversity. And secondly, potential future uses include personalized medicine. Okay, and this is not that far out. I would not be surprised if within 10 years, we're at this point where individual humans have their entire genome sequenced, and we use that genetic information to predict health problems and prescribe appropriate drugs and treatments for that individual person. Because depending on your genes, you might need a different dosage of a drug, or you might need a different drug altogether compared to someone else that has a different gene. And so this could be potentially groundbreaking in being able to treat people in an individualized way and potentially save many lives. We are now at the end of SL content. We do have just a little bit more of HL content, so stick around if you're HL. SL students, you're done with this subtopic. Difficulties applying the biological species concept to asexually reproducing species and to bacteria that have horizontal gene transfer. The biological species concept doesn't work well with groups of organisms that don't breed sexually or where genes can be transferred from one species to another. So if we're saying that a species is a group of organisms that can breed and produce fertile offspring, what happens if they're asexually reproducing? They don't require 2 individuals. They basically just duplicate themselves exactly. Well, that can be really hard to then define what is a species because we can't use that definition. Examples of this are blackberries and dandelions. They only reproduce asexually. Their offspring are produced from replicating their cells exactly, so they're genetically identical to the parent plant, known as clones. If a clone doesn't interbreed with other clones, According to the biological species concept, it's its own species. So blackberries and dandelions who have abandoned sexual reproduction are no longer species according to the biological species concept, right? So that is one difficulty to apply this definition to this particular group of organisms. Next difficulty are species that have horizontal gene transfer. This is when we have genes transferred between two different species. Now, this is very frequent among bacteria. For example, they could horizontally transfer a gene for antibiotic resistance between species. In fact, there is so much gene transfer between bacteria that it is debatable whether the biological species concept works at all with prokaryotes. Chromosome number as a shared trait within species. Remember, eukaryotic body cells contain 2 of each type of chromosome. You have 23 pairs, 23 from mom and 23 from dad. For sexual reproduction to occur, males and females of the species divide these chromosomes in half in a process called meiosis, which we'll learn about in the future, to produce those haploid gametes, like eggs and sperm, that will just have 1 copy of each of their chromosomes. So instead of 23 pairs, like in every cell in your body, egg and sperm cells will just have 1 of each. So these will get divided in half and split as gametes are produced. And so the benefit to this is that male and female gametes then fuse to produce a diploid zygote, that fertilized egg. And so if you have 2 halves, you can then fuse to form a whole again. The 1 set of chromosomes in the male gamete plus the 1 set of chromosomes in the female gamete now form offspring that have the same chromosome number as the parent. If 2 organisms with different chromosome numbers mated, the chromosomes wouldn't be able to pair up and fuse to form a zygote, and the resulting cells would not be viable. This is exactly what happens with a horse and a donkey. The mule has then an odd number, and so it cannot pair up. Crossbreeding between closely related species is unlikely to produce fertile offspring if the parent chromosome numbers are different. Next, you do need to be able to engage with local plant or animal species to develop a dichotomous key. We are doing this by filling out this classification of the dichotomous key activity. You will need to work through this and practice using a dichotomous key, and you'll even be making a dichotomous key to demonstrate your understanding of how they work. Lastly, identification of species from environmental DNA in a habitat using barcodes. DNA barcodes are short sections of DNA from one gene, or at most several genes, which are distinctive enough to identify a species. So it's just a short section of a gene that is distinct to a species. DNA barcoding allows scientists to identify species from small pieces of tissue that might otherwise be difficult to recognize by comparing the DNA barcode to a reference library. We can use this for species identification from environmental DNA collected from water, soil, or any other part of the abiotic or non-living environment. Typically, these samples contain DNA from a wide diversity of organisms that have interacted with the sample environment, and we can use DNA barcodes to identify these organisms. This method allows the biodiversity of habitats to be investigated rapidly. So we can take a water sample or a soil sample, and we can run this DNA barcoding and be able to identify what species are present in that location. All right, that's it for A3.1. Good luck out there.